Hello everyone and welcome back. Let's go ahead and move on in our discussion of genetic material that we find within cells. Now we are going to go past DNA though and start talking about RNA. RNA fills a lot of roles in the cell. Remember, RNA is special because it can travel throughout the cell, right? Unlike DNA, which in eukaryotic cells is confined to that nucleus. So in this course, you are expected to know three main types of RNA. Now, keeping in mind that there are actually a lot of other types of RNA in a cell, these are the three main types that we are going to focus on in this course specifically, okay? And that's mRNA, tRNA, and rRNA. Sound good? So let's go ahead and start with mRNA. So mRNA, the M standing for messenger RNA, is so named because it basically acts as the messenger of sorts between DNA and protein productions, right? It carries the message that DNA provides out of the nucleus and to the ribosomes where proteins can be made. During a process called transcription, DNA is transcribed into mRNA. This mRNA is then translated into protein. So mRNA serves as the genetic template of a protein since the DNA can't leave the nucleus. Next, we have tRNA, or transfer RNA. This is possibly one of the coolest forms of RNA, so I'm happy that we get to go over it in this course. And one of the reasons why this is so cool is because RNA is generally, as we know, single-stranded, right? DNA is usually double-stranded, RNA single-stranded. But tRNA is kind of cool because even though it is technically still single-stranded, it folds back on itself to form this distinct T-like shape, as you can see here. So without going too far into it, transfer RNA is responsible for bringing amino acids together during translation to form the growing polypeptide chain that will become a protein. So it contributes to the steps that mRNA has previously contributed to as well, right? mRNA is the message that gets read, and tRNA helps with that reading so that way we can make those proteins, all right? And now there is actually another form of RNA that also contributes to the synthesis of proteins, and that is the last form of RNA that you will learn in this lesson, and that is rRNA. rRNA being ribosomal RNA. This RNA is a main component of ribosomes, which we have gone over previously when we were looking into the different parts of the cell. Now, rRNA combines with special proteins to form ribosomes, right? That's what a ribosome is actually made out of, is proteins and rRNA. And ribosomes are responsible for quote-unquote reading mRNA to form proteins. And once they have read that mRNA, it's the tRNA that transfers amino acids to the ribosome to help build that polypeptide. So all these work together to help contribute to the cell's function. We have the genetic material, and that genetic material is read and transcribed into messenger RNA. That messenger RNA is then read by ribosomes, and ribosomes being made up of rRNA and proteins. And mRNA is then interpreted or translated into proteins with the help of transfer RNA, because the transfer RNA is what brings those amino acids to that ribosome one at a time to fulfill that role. Does that make sense? So circling back to rRNA specifically, ribosomes contain rRNA to help build proteins. So bringing all this together, sections of DNA are transcribed into mRNA, which is brought from the nucleus to the ribosome, which is made up of rRNA and proteins. And the mRNA is then read by the ribosome, which brings the tRNA in with the appropriate amino acids to build whatever protein the mRNA is a template for. Make sense? Okay. Now, I find that a lot of students benefit from learning comparatively. So let's go ahead and do a really quick review on comparing and contrasting RNA to DNA. Sound good? So nice and quick. RNA and DNA are both found in all living cells. For it to be a living cell, it must have DNA and RNA. It must have genetic material, right? Now, breaking it down more specifically, in eukaryotic cells, DNA is only in the nucleus. It cannot leave that nuclear space, which is why messenger RNA is so important. So, in eukaryotic cells, DNA is in the nucleus only. However, RNA in eukaryotic cells can be inside or outside of the nucleus. It can be just about anywhere in the cell. In contrast, prokaryotic cells have no organelles. And so we can find DNA and RNA in specific regions, maybe of the cell. For instance, we say DNA is found in the nucleoid region, but there's no membrane-bound organelles to confine DNA and RNA. Similarities again, both are obviously nucleic acids, right? So their names are deoxyribonucleic acid and ribonucleic acid, DNA, RNA, meaning both must be built of monomers that are called nucleotides, and both have the same fundamental structure in their nucleotides, right? The nucleotides of both are made up of 3 parts: a phosphate group, a sugar, and a nitrogenous base. However, where they differ is in those sugars and a little bit in those nitrogenous bases. Number 1, the sugars in DNA, or the sugar that's used in DNA monomers, is deoxyribose sugar. So it's missing the oxygen, the sugars that go into making RNA has. So deoxyribose sugar is what contributes to a monomer of DNA. And that's why we have the name deoxyribonucleic acid, right? DNA. In contrast, RNA is made up of a ribose sugar, hence the name ribonucleic acid. Now, the nitrogenous bases between these two are pretty similar. We see some similarities and one minor difference, right? In DNA, guanine and cytosine are nitrogenous bases that are complementary to each other, so they are always paired together in our double strand, right? And we also have adenine, which complementarily pairs with thymine, right? So our 4 bases are guanine, cytosine, adenine, and thymine. But in contrast, RNA has guanine and cytosine as well, and again, those are base pairs, they pair together. But RNA, instead of thymine, has something called uracil. So adenine actually binds with uracil in RNA because there is no thymine in RNA. Sound good? Okay. Now, looking at structure and size, DNA is usually double-stranded, while RNA is usually single-stranded. And DNA is much, much longer than RNA because DNA must code for all the proteins that the organism has or needs, right? This is in stark contrast to RNA, which does not need to fulfill that role. So DNA has to code for literally everything, and so it is huge compared to RNA. All right, so hopefully that was a helpful overview. Just to make sure that we're all on the same page, I'm just going to go over 3 quick practice problems with you all. Sound good? All right, so with that, let's practice. Let's start with looking at a segment of DNA and fill it in together. Sound good? So if one DNA strand has the bases shown on the screen, what would the other strand of DNA look like? All right, so that's just working on practicing base pairing within DNA. But now, this next question, let's look at how it would look if we were to practice transcription and look at how the complementary set of RNA would look to DNA. So now we're practicing basically writing messenger RNA. Sound good? So in a process known as transcription, an enzyme transcribes DNA into mRNA by complementary pairing RNA nitrogenous bases to a DNA template strand. If this is the DNA template strand that you see on the screen, what would the complementary mRNA strand look like? Okay, and now last but not least, I want you to fill in the following blanks to summarize the relationship between DNA, RNA, and proteins. I've provided a little word bank over here for you to choose from to fill in these following blanks. Sound good? Okay. And to keep the different blanks straight, I'm going to replace the blanks with A, B, C, and D. Sound good? And definitely keep in mind that the words in the word bank can definitely be used more than once. So, A is the genetic material that ultimately codes for all proteins in a cell. In the nucleus of a cell, A is transcribed into B by a protein called RNA polymerase. Once transcribed, B leaves the nucleus and is taken to a ribosome. A ribosome which is made up of C and special proteins. The ribosome then reads the B and translates this into proteins. Proteins are made up of amino acids bonded together. D brings or transfers amino acids to the ribosome so the amino acids can be bonded together according to the mRNA sequence, eventually forming a functional protein. Well, I hope that this was helpful for you all, and always feel free to reach out with any questions, comments, or concerns, and try to make sure that before moving on to your next step in this lesson series, you fully understand and grasp the concept of DNA and RNA, at least these 3 types of RNA, and that you can understand their role and overarching picture in their contribution to life and within the cell. Sound good? Okay, and with that, best of luck.